Hello everyone, welcome to my channel Anupma Biology classes. In this video, our topic is Principles of Biotechnology. So from this video, we will start a new series of biology, that is biotechnology. This video is extracted from CBSE class 12th, part 1st of chapter 11. This series is very important from examination point of view and interesting also. So let's begin the lecture. Here you can see the lecture overview. So in this video, we will see what is biotechnology, introduction and definition of biotechnology, principles of biotechnology, conceptual development of the principles of genetic engineering, construction of the first artificial recombinant DNA molecule, and recombinant DNA technology. So this is the lecture overview of this video. Now, here the first thing we have to know that what is biotechnology? So, manipulation of gene and genetic material by man is a fast emerging branch of science which started with the formation of recombinant DNA molecules. This branch of science is named as recombinant DNA technology, genetic engineering, and DNA manipulation biotechnology. Now, here you can see that there are many branches of the biotechnology, or fields of the biotechnology, that is agriculture, nutrition, health and medicine, industrial, bioweapons, environment, arid, marine, bioinformatics, and ethics and laws. Now, what are the scope of biotechnology? So here you can see the biotechnology industries like pharmaceuticals, medical devices and equipments, digital healthcare, now information technology in which the telemedicine, quantum computing, biological computing, optics, facilities and infrastructure management, nanotechnology, chemicals, that is protein and peptides, molecular electronics, industrial biotechnology and agriculture. So these are the industries or scope of biotechnology. So, biotechnology is a multidisciplinary pursuit that has emerged as a demanding industry during the recent past. Besides being a branch of advanced biological science, it has attracted many multinational companies, including those are concerned with the production of pharmaceutical products for the cure or control of many human diseases. These products include antibiotics, vaccines, life-saving drugs, and gene therapy. Then improvement of clinical testing and diagnostic tools and the production of novel varieties of crop plants and animals are also the scope of biotechnology or use of biotechnology. Now the question is, what is biotechnology? A general description of biotechnology is using living things to create products or to do tasks for human beings. Biotechnology is the practice of using plants, animals, and microorganisms such as bacteria, as well as biological processes such as the ripening of fruits or the bacteria that break down compost to some benefit. For example, biotechnology is used in industry, medicine, and agriculture to produce foods, medicines, test for disease, and remove waste. It can also be used to solve problems and conduct research. Over time, biotechnology has formed the basis of learning about people and disease. Biotechnology has also underpinned the development of treatments. So, this is biotechnology. Now, why do we do biotechnology? So, biotechnology is used in a wide range of applications in food, science, medicine, the environment, and agriculture. Research is rapidly expanding the possibilities of where it will be used next. Any technology brings with it risks as well as benefits, and gene technology is no exception. These risks need to be careful assessment before a genetically modified plants, animals or microorganism is released. Government regulatory authorities assess the risk, which may include how readily the released organism could crossbreed with similar organism in the environment, whether the modifications gives the organism extra survival advantages, whether these advantages could upset a balanced ecosystem. So, this technology, that is recombinant DNA technology, involves cutting and pasting of desired DNA fragments into the specified host for the benefits of human beings. This branch of science is based on the following important discoveries. So, what are the discoveries in the DNA manipulation biotechnology? So, occurrence of plasmids in bacteria which replicate along with the independent of bacterial chromosomal DNA. An observation in early 1960s that the DNA of bacteriophage is broken down into pieces when it infects a bacterium. A conclusion was drawn from this observation that the bacterial cells have some defensive mechanism that break the viral DNA. This observation led W. Arbour to postulate that the bacteria must be having some enzymes which can break the viral DNA. Arbour, Nathan and Hamilton Smith in 1970 isolated first restriction endonuclease from the bacterium Haemophilus parainfluenzae, which can break DNA at specific sites, and for this awarded Nobel Prize in 1978 also. These enzymes are called molecular scissors. Genetic engineering was started by Paul Berg in 1972, who introduced a gene of SB40 into a bacterium with the help of lambda phase. Berg was awarded Nobel Prize in 1980 and considered as father of genetic engineering. Annie Chang and Stanley Cohen in 1973 generated first recombinant DNA molecule by combining a gene from a bacterium with a plasmid of E. coli or Escherichia coli. Collins and Horn in 1979 developed a method for obtaining recombinant plasmids packed into the protective coats of lambda phase and then transduced into E. coli. So, this is a normal and general view of biotechnology. Now, after this, introduction and definition of biotechnology. So, according to introduction, biotechnology is the application of science techniques to modify and improve plants, animals, and microorganisms to enhance their value. Biotechnology allows for the manipulation, synthesis, and eventual creation of genes. Genes are the encoded roadmap for an organism development. Thus, by manipulating genes, agricultural biotechnology are able to engineer the plants they desire down to the fruits. So, according to the name, that is biotechnology, it is formed by the two words. First one, that is the biology, that is the study of science of living things, and second is technology, that is the use of science in the solving problems or the making of products which are beneficial to mankind. So, by these two words, the new name created, which is biotechnology. So, biotechnology is the combination of biology and technology. Any technique that uses any organism or part of organism to make or modify product is biotechnology. The origin of biotechnology is an old as human civilization. As a science, it is a little more than 100 years old. As an old useful art, it dates back into antiquity. Fermentation is the oldest biotechnological process discovered by people by prolonged soaking of grains before cooking or by storing juices of fruits or palms. Fermented honey or meat may have provided one of the earliest alcoholic beverages fermentation for man. Other early fermentation process practiced by man included leaving of bread, rating of flax, production of alcoholic beverages, production of vinegar, production of curd, etc. So, biotechnology provides us with a wide range of products in considerable amount. A number of powerful industries based on beneficial activities of microbes have been set up in the recent years to produce a variety of products in enormous amount. Here you can see some of these products like food items, that is bread, idli, dosa, etc. Dairy products like yogurt or curd, cheese, butter, kefir, etc. Alcoholic beverages like beers, wines, whiskey, etc. Non-alcoholic beverages like curing of coffee beans and tea leaves. Organic acids like the lactic acid, citric acid, acetic acid, gluconic acid, etc. Enzymes, vitamins, hormones, amino acids, antibiotics, vaccines, steroids, etc. So these are the products by biotechnology. During the last 2 decades, biotechnology has achieved greatest achievements in the field of recombinant DNA technology, that is polymerase chain reaction (PCR), cell culture and fusion, bioprocessing, gene cloning, DNA fingerprinting, in vitro fertilization leading to a test tube baby, developing a DNA vaccine, correction of a defective gene, etc. Owing to the multifarious developments in the field of biotechnology and its achievements, the term biotechnology is currently used in a restricted sense to refer to only those processes which are concerned with the genetic manipulation to achieve the same end on a large scale. A recent definition of the biotechnology given by European Federation of Biotechnology, that is EFB, combining both the traditional view as well as the modern molecular biotechnology, is as follows. That is, biotechnology is the integrated use of biochemistry, microbiology, and engineering science in order to achieve technological applications of the capabilities of microorganisms, culture tissues and cells, and parts thereof. One time, I will repeat it once again. That is, biotechnology is the integrated use of biochemistry, microbiology, and engineering science in order to achieve technological applications of the capabilities of microorganisms, culture tissues or cells, and parts thereof. So, this is the introduction and definition of biotechnology. After this, what are the principles of biotechnology? So, the science of biotechnology is based mainly on the two core technologies, and these are: first one, that is genetic engineering, and the second is biochemical engineering. So, what is genetic engineering? Genetic engineering is the manipulation of genes by man. Genetic engineering refers to artificial synthesis, isolation, modification, combination, addition, and repair of the genetic material, that is DNA, to alter the phenotype of the host organism to suit human needs. And the second is biochemical engineering. What is this? Biochemical engineering process that helps the growth of desired microbe or eukaryotic cell in large quantities in a sterile medium, that is tissue culture technique, for the manufacture and manipulation of biotechnological products, that is antibiotics, vaccines, enzymes, medicines, hormones, etc. Now, after this, conceptual development of the principles of genetic engineering. So according to concept, here you can see the genetic engineering, the technology entailing all process of altering the genetic material of a cell to make it capable of performing the desired functions such as producing novel substances. In other words, genetic engineering is the deliberate, controlled manipulation of genes in an organism in order to upgrade that organism. In genetic engineering, recombination can also refer to artificial and deliberate recombination of pieces of DNA from different organisms, creating what is called recombinant DNA. So here you can see the applications of the genetic engineering, that is farming. Gene farming is a technology that scientists use to alter an animal's own DNA or to splice in new DNA called a transgene from another species. In farming, these genetically modified animals are mostly used to make human proteins that have medicinal value. The proteins encoded by the transgene is secreted into the animal's milk, eggs or blood and then collected and purified. So this is the application of genetic engineering. So according to concept, the living organisms reproduce mainly by the asexual and the sexual methods. Out of the two, sexual reproduction is more advantageous as it provides opportunities for the variations and formation of new genetic combinations. Asexual reproduction, on the other hand, preserves the genetic characters. The variations produced a result of a sexual production may be beneficial to the organism as well as populations. So, this hybridization procedure or beneficial activity of the sexual reproduction is useful in plant and animal breeding programs where hybridization procedure for the combination and multiplications of the desirable genes. However, traditional hybridization procedures very often include and multiply some of the undesirable genes along with the desired ones. So, to avoid the inclusions of the undesirable genes, the new technique of genetic engineering has been developed which involves creation of recombinant DNA, use of gene cloning and gene transfer. As compared to traditional hybridization method, the technique of genetic engineering is referred as it permits to isolate only the desirable genes and introduce it into the target organism. This new technique excludes the introduction of undesirable genes into the target organism. Now, let us discuss the fate of the piece of DNA which has been introduced into the alien, that is foreign organism. This piece of DNA would be able to multiply itself in the organism. It can multiply only when it gets integrated into the genetic material of the recipient organism. This is because the foreign piece of the DNA has now become the part of a recipient chromosome which possesses the ability to replicate. Normally, a specific DNA sequence present in the chromosomal DNA called the origin of replication is responsible for initiation of replication. If this sequence is present, the DNA will replicate. If it is absent, the DNA will not replicate. Therefore, the alien piece of DNA fails to replicate until it gets integrated with the chromosomal DNA having origin of replication. In this way, the alien DNA linked with the origin of replication replicates and multiplies in the host organism. This is called the cloning, that is formation of multiple identical copies of any template DNA. So this is the conceptual development of the principle of genetic engineering. After this, how the first artificial recombinant DNA molecules formed. Before this, you can see here that construction of the first artificial recombinant DNA molecule, that means the way by which the artificial DNA molecule will form. So, the first artificial recombinant DNA molecule was constructed in the following manner. In the year 1972, Stanley Cohen and Herbert Boyer, the two scientists of USA, isolated an antibiotic resistant gene from a plasmid of the bacterium Salmonella typhimurium. This piece of DNA was responsible for conferring antibiotic resistance. These two workers isolated the antibiotic resistance gene by cutting the desired piece of DNA from the plasmid. Cutting off a piece of DNA from a plasmid was done with the help of restriction enzymes. popularly known as molecular scissors. The piece of DNA cut from the plasmid was then linked with the plasmid DNA, acting as vector. Linking of the fragment of DNA with vector was done with the help of another enzyme, that is the DNA ligase, which acts on cut DNA molecules and join their ends. This newly formed DNA having integrated fragments of the antibiotic resistant gene is called recombinant DNA. The vectors used to transfer recombinant DNA to the bacterium E. coli, this transfer of recombinant DNA is exactly similar to the transfer of malarial parasite from diseased person into the healthy human body through mosquito. The vector acts as a mosquito in transferring the piece of DNA from donor to the recipient organism. Once the newly formed recombinant DNA was transferred to bacterium E. coli, it replicates in the host cell by using the enzyme DNA polymerase. It results in the formation of several copies of recombinant DNA having capability of the antibiotic resistance. The process of manipulation, multiplication of genes, that is pieces of DNA, is called cloning. It results in the formation of several copies of antibiotic resistant gene in E. coli or cloning of antibiotic resistant gene in E. coli. So this is the constructional way for the first artificial recombinant DNA molecule. So here you can see the first artificial DNA molecule. Here you can see the process of recombinant DNA technology. So in the process of recombinant DNA technology, here the gene for antibiotic resistance here enter by the restriction enzyme which cut and join it. Then the DNA ligase is responsible for the joining of this and formation of new plasmid. So by this the modified new plasmid will be formed. So in this picture you can see the restriction enzyme which cut at the specific point, which is the gene of interest. Then this gene is inserted into a new antibiotic resistance gene, and this addition which help of enzyme by the DNA ligase and formation of the modified plasmid. So cloning or expression vector, that is the plasmid, here you can see in the other side eukaryotic chromosome. Then by this in eukaryotic chromosomes DNA fragments containing gene of interest, then digestion with the restriction endonuclease. By this then new plasmid added with the gene of interest, and after this a recombinant plasmid will be formed. So according to the birth of recombinant DNA technology, in the year 1972, D.A. Jackson, R.H. Simmons and Paul Berg reported the construction of first recombinant DNA under in vitro conditions in a test tube. The constructed recombinant DNA molecule consisted of SB40 DNA, bacteriophage lambda genes, and the galactose operon of E. coli. For this discovery, Paul Berg was awarded Nobel Prize in Chemistry in 1980. In 1973, S. N. Cohen, A. C. Y. Chang, H. W. Boyer, and R. B. Helling inserted kanamycin resistance gene of E. coli in the plasmid PSC101 and introduced the recombinant plasmid in the E. coli cells. Hence, they were able to obtain multiple copies of kanamycin resistant gene. This was the first successful gene cloning experiment. For this discovery, Stanley Cohen was awarded Nobel Prize in Physiology or Medicine in 1986. So the history of recombinant DNA technology in 1976, Herbert Boyer co-founded Genentech, the first firm founded in the United States to apply recombinant DNA technology. In 1978, somatostatin, which regulates human growth hormones, is the first human protein made using recombinant technology. So here, if we talk about the whole process of the recombinant DNA technology, it refers to the joining together of the DNA molecules from the two different species that are inserted into a host organism, and the two different species here a production of a new varieties. So, in the process of recombinant DNA molecule, which are the DNA molecules formed by laboratory method of the genetic recombination to bring together genetic material from the multiple sources, creating sequences that would not otherwise be found in the genome. So this is the process of recombinant DNA technology and the formation of recombinant DNA molecule. Now, after this, what is recombinant DNA technology? So if we talk about the recombinant DNA technology, here you can see that here the donor cell in which the foreign DNA, which with the help of restriction enzymes cut at specific sites. Then here on the other side bacterial cells where the plasmid, that is vector DNA, present, and here the ligase enzyme joins foreign DNA to the plasmid. After this the formation of recombinant DNA, then the host bacterium with the recombinant DNA, then formation of clones of rDNA and the clones of engineered bacteria in a medium, then recombinant product and downstreaming occur, that is separation and purification of product. So this is the whole process of the recombinant DNA technology and the diagrammatic representation of the recombinant DNA technology. Recombinant DNA technology, popularly known as genetic engineering, is a stream of biotechnology which deals with the manipulation of genetic material by man in vitro. It refers to a number of methodologies in which the desired genes or DNA sequences of an organism are cut into fragments and then introduced into host cells with or without the help of carriers or vectors to alter its phenotype to suit human needs. Now, techniques for introducing foreign genetic material into plant cell genome. So, what are the techniques? The first technique is here, that is through a vector, and the second technique is through direct introduction of DNA. So, in first process, that is through the vector, here the selection and isolation of the desired fragments of the DNA which containing gene sequence that needs to be cloned, known as insert. Then the generation of recombinant DNA molecule by insertion of these inserts, that is DNA fragments, into the carrier DNA molecule termed as vector. And then after this, here the introduction of the rDNA molecules into the host cell, and this process here the recombinant DNA formed by the vector plus insert. Now, if we talk about the second, through introduction of DNA. So, through introduction of DNA, here first process, that is co-cultivation, which you can see in this picture, that is culturing the recipient protoplast with purified DNA. After this, the second process, that is electroporation. We will discuss each and every part in detail in the next video. That is application of the electric impulses to change the porosity of protoplasts so that they directly imbibe the purified DNA. And the next is and last is microinjection, that is direct injection of DNA fragments with the help of micropipette. Other methods of the gene transfer are liposome mediated gene transfer, calcium phosphate precipitation method, transformation of by ultrasonification, and transformation using pollen or pollen tubes. So, thus genetic engineering can be defined as the generation of new combinations of the heritable material by the insertion of desired genes or DNA sequences produced by whatever means outside the cell into any carrier system so as to allow their incorporation into a host organism in which they do not normally occur but in which they are able to perform normal behavior and propagation. So this is the end of the video in which we understood about the process of biotechnology, introduction and definitions of biotechnology, principles of biotechnology, and recombinant DNA technology. After this, the next upcoming video in the biotechnology series will be on the tools of recombinant DNA technology. So if you understand this video, please Please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.